Hello guys, welcome to Edudextro. I am Mohamed Irfan. In this video we are going to be doing the very first lecture of the class 12 zoology chapter, that is biotechnology principles and processes, and we will be doing NCERT line by line. So without any delay, let's go into the academics. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. So this is a kind of primitive form of the definition for biotechnology, wherein we use live organisms like microbes and the enzymes which are produced from these organisms to produce some products and some processes which are useful to humans. Okay, in this sense, making curd, bread or wine, which are all microbe mediated processes, could also be thought as a form of biotechnology, right? We are making curd with lactobacillus, we are making bread with Saccharomyces, right? And even wine, we are using microbes for making wine. So all these processes are considered part of biotechnology, wherein we use the organisms themselves or the enzymes from these organisms. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. So now it is not only these microbes and their enzymes which are used in biotechnology, but there are genetically modified organisms which are used according to our own needs to produce the product in a larger scale. Okay, so even many other processes are also involved today under biotechnology. For example, IVF, that is in vitro fertilization, which leads to a test tube baby, is also considered under biotechnology, and synthesizing a gene and using it is also considered under biotechnology. Developing a DNA vaccine is also biotechnology, and even correcting a defective gene is also part of biotechnology. So it is not only the primitive idea that we are using microbes and their enzymes for our products like food, but even other processes are part of biotechnology. Now we have a very good and precise definition given by the EFB, that is the European Federation of Biotechnology, that is EFB. They have given a definition of biotechnology which not only accommodates the traditional view of biotechnology but also the modern molecular biotechnology. So this is the definition which has been given by the EFB: the integration of natural science and organism. Okay, organisms themselves, the science behind these organisms, the cells of these organisms, the parts of the cells of these organisms, and molecular analogues, meaning even molecules from these organisms, when all these are used for products and processes and services, that is called biotechnology. So this is the definition given by the EFB. So this is the next topic, that is principles of biotechnology. So there are two core techniques that are involved in modern biotechnology, and these are the fundamental techniques of biotechnology. So one is genetic engineering and the other one is called bioprocess engineering, and that word is not given in NCERT, but let's note it down here that the other process is called bioprocess, sorry for my handwriting, but yes, bioprocess engineering. Now this point stands for bioprocess engineering. Sorry for my handwriting, will make it better soon. So what is genetic engineering? This is a engineering wherein we alter the chemistry of the genetic material, let it be a DNA or a RNA. So we are causing some chemical changes in the genetic material, that is DNA and RNA. So that is simple, guys. What do you mean by a chemical change in the genetic material? Basically we are doing some changes in the nucleotide sequence, and that is what we call as a alteration in the chemistry of the genetic material, and these are introduced into the host organisms, and thus it will cause a change in the phenotype of the host organism. Now the second process, that is bioprocess engineering. It is basically a engineering wherein we maintain a sterile ambience. Now sterile ambience means a environment wherein there should not be any microbial contamination. Now over here, contamination means if you want to grow E. coli for example, you should not have any other microbe growing in that environment, and that is accomplished using bioprocess engineering. Maintenance of sterile ambience in chemical engineering process to enable growth of only the desired microbe or the eukaryotic cell in large quantities for the manufacture of the biotechnological products like antibiotics, vaccines, enzymes, etc. So you can understand how crucial bioprocess engineering will be in the manufacture of a vaccine or antibiotic, right? So guys, now let's understand the concept behind the development of the principles of genetic engineering. We already know the advantage of sexual reproduction over asexual reproduction. We already know that sexual reproduction can bring about variations, and it can bring about unique combinations of genetic setup by bringing genes from two different individuals and forming a new combination. And we already know that how much beneficial it is for the organism itself, and how much beneficial it is for the entire process of population and evolution, right? But asexual reproduction can preserve the genetic information, right? But sexual reproduction permits variation. Now, the traditional hybridization procedures used in plants and animal breeding, it leads to inclusion and multiplication of undesirable gene, and this is called inbreeding depression. While it can also bring about the combination of desired genes as well, or it can also give rise to a condition wherein both desired and undesirable genes are there. Now the techniques of genetic engineering includes the creation of a Rdna, which full form is recombinant DNA. Okay, so the short form of recombinant DNA is called Rdna, using gene cloning and gene transfer, and this helps us to overcome this limitation, and it allows us to isolate and introduce only one or a set of desirable genes without introducing the undesirable genes into the target organism. So this technique of genetic engineering, wherein you can create a recombinant DNA, you can create your own combination using gene cloning and gene transfer. Gene cloning is basically creating many copies of what you have created and then transferring it. Using this, you can overcome the limitation, and you can introduce and isolate only the desirable genes without introducing the undesirable genes into the target organism. So we can overcome the problem of inbreeding depression. So guys, do you know what exactly will happen if you just introduce a piece of DNA just like that, without any genetic engineering? If you are introducing a piece of DNA just like that into an alien organism or into the cell of the organism, you know what exactly will happen? There won't be anything. If you want that piece of DNA to divide, you can't expect that to be in the progeny of the cell of the organism if you are just introducing the gene in a, you know, just like that. If you're introducing, you cannot expect that to be in the progeny. So let me draw what I am telling right now. So this is your cell having the genome in yellow color. Now if I want to introduce a red color gene, and if I simply put it just like that into the cell, and if this cell divides into two daughter cells, the end point will be that it will just have these two yellow genes. It won't have the red gene, because we have introduced that red gene without any knowledge of genetic engineering. We can't expect to see that red gene in the progeny of the organism. Okay, but but when it gets integrated into the genome of the recipient, if I insert this red gene, if I combine it with the yellow gene, if I insert it in between of the yellow gene, then it may multiply and be inherited along with the host DNA. Okay, so if this is the other case wherein there is a genome, and if I introduce my gene somewhere in the middle or somewhere in the host genome, then when that divides to give progeny, then at that time I can expect the red gene in the new genome. Okay, so if this is the daughter cells, I can expect the red gene also within those cells. So this is the essence of genetic engineering. This is because the alien piece of DNA has become part of a chromosome which has the ability to replicate. Okay, so unless and until you haven't caused changes in the genetic makeup of the cell, you cannot expect a change in the genetic makeup of the progeny. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. So within a chromosome, there is a specific DNA sequence which is called the origin of replication, or the ORI. Okay, this is called ORI. Sorry for the handwriting. All are in small letters. Let me write it a little more neater. Yes, all are in small letters. Okay, O, R, and I. ORI, which is responsible for initiating the replication. Therefore, for multiplication for any piece of DNA in an organism, it needs to be a part of a chromosome which has a specific DNA sequence known as the ORI. So unless and until you are introducing your gene into a chromosome which is having a ORI, then only you can expect a foreign gene even in the daughter cell. So what does this mean? This cell has ORI as well. That is why I am able to see my foreign gene in the daughter cells as well. Okay, thus an alien DNA is linked with the ORI so that this alien piece of DNA can replicate and multiply itself into the host organism. And this can also be called as cloning, or making multiple identical copies of any template DNA. Okay, so you are making multiple copies. Now if this divides further, if this continues further, you are getting multiple copies, right? You are getting multiple copies. So this is called cloning. Okay, Now let us focus on the first instance of construction of an artificial rDNA molecule. Now we are going to see how was the first rDNA molecule. I have already said what is recombinant DNA molecule. We have just made a recombination of DNA. We have just introduced a chemical change. We have just introduced a foreign piece of DNA within a larger DNA, that is called the recombinant DNA. So now let's see how this construction took place. So the construction of the first rDNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. So we have linked a gene which encodes for antibiotic resistance with a native plasmid. Now what is plasmid? Plasmid concept is very important. It is a autonomously replicating circular extrachromosomal DNA. Autonomously. Autonomously means by itself it can replicate. By itself it can replicate. It is circular. It is extrachromosomal. Means it is apart from the chromosome. It is a piece of genetic material and it is a DNA. So if this is your cell, consider this as your cell, okay? Then apart from your genetic material, there is a circular structure called this plasmid. Okay, so this is the plasmid, and the possibility arised when we linked a gene which encodes the antibiotic resistance with this native plasmid. This native plasmid is of Salmonella typhimurium. So this is a bacteria called Salmonella typhimurium which has a plasmid, and when we linked the gene encoding antibiotic resistance, that gave the first construction of the rDNA. So Stanley Cohen and Herbert Boyer were the two scientists who accomplished this in the year 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring the antibiotic resistance. So Stanley Cohen and Herbert Boyer in 1972, they were able to isolate the specific gene, the piece of gene from a plasmid, and this region of the plasmid was responsible for conferring the antibiotic resistance. The cutting of DNA, the cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissors or the restriction enzymes. So, as I said, there was a plasmid, right? There was a plasmid from which they had cut out this region, right? From this larger plasmid, they had cut out this region which was responsible for the antibiotic resistance. And the cutting out of this piece was accomplished using molecular scissors or the restriction enzymes. Scissors, they are able to cut the molecules, and this is called the restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. So this blue color piece of DNA was then linked with another plasmid DNA. So these plasmid DNA again, which plasmid DNA? The other plasmid DNA act as vectors to transfer the piece of DNA attached to it. So this piece of DNA wherein you will be attaching the foreign piece of DNA is a vector. Just like how parasite, maybe if you consider the malaria causing plasmodium, just like how it gets attached to the mosquito and how the mosquito acts like a insect vector, just like that, if you consider your foreign gene or your requisite gene as a, you know, a malaria causing plasmodium, then the mosquito acts like the plasmid. Okay, so this is how we can relate this with the malaria plasmodium analogy. So the linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase. So till now we have seen two enzymes, that is restriction enzymes and DNA ligase. So DNA ligase is a enzyme which can stick together two ends. Okay, if there are two ends like these, and if you have a foreign gene, then this can be stuck with this using DNA ligase, and over here this can be cut using restriction enzymes. Okay, so basically both are antagonistic enzymes, right? So this acts on DNA molecules and joins their ends. This makes a new combination of circular autonomously replicating DNA, right? Plasmid, right? Circular autonomously replicating DNA created in vitro. In vitro means in the lab, right? This means in lab. Sorry for handwriting. And it is known as the Rdna or the recombinant DNA. So thus, using restriction enzymes, we have cut the piece of DNA. Using DNA ligase, we have stuck it, and thus we have made the recombinant DNA in vitro. We have made the recombinant DNA successfully. When this DNA is transferred into E. coli, that is Escherichia coli, and this is a bacteria which is closely related to Salmonella. So though both are not the same bacteria, they are kind of closely related to Salmonella. That is why we are introducing the plasmid from Salmonella to E. coli, right? Then it could replicate using the new host's DNA polymerase enzyme. So using the specific mechanisms of the host, the foreign gene is replicating. That is what we had told 2 to 3 slides behind, right? So using the DNA polymerase enzyme, it is able to make multiple copies. The ability to form multiple copies, as we know, is called cloning in E. coli, right? And we are cloning the antibiotic resistance gene in E. coli, right? You can hence infer that there are three basic steps in genetically modifying an organism. Firstly, we have to identify the DNA with the desirable gene. Secondly, we have to introduce this DNA into the host, right? And thirdly, we have to maintain the introduced DNA in the host and the transfer of the DNA to the progeny. So this is what we have discussed so far, and these are the three basic steps required in genetically modifying an organism. Okay, so now we are going to see the tools of Rdna technology. Now, till now, what all we have discussed about genetic engineering or Rdna technology, it can be only accomplished using a restriction enzyme. It should have a polymerase enzyme, it should have ligase, and it should have vectors. These are the four terms that we have come across till now, and yes, of course, the host organism as well. And these five things are the key members of our DNA technology. Now let's see some of these in detail. NCERT is explaining some of these in detail. So firstly, let's see what is restriction enzyme. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage. Now what is bacteriophage? Bacteriophage is the virus which affects bacteria in E. coli were isolated. Okay, so in 1963, two of these enzymes which were responsible for preventing the growth of bacteriophage were isolated. That is why they have been named restriction enzymes. Though they are molecular scissors, they have been named as restriction enzymes because they restricted the growth of the bacteriophage in E. coli. One of these added methyl groups to the DNA while the other cut the DNA. The latter one was called the restriction endonuclease. Endo means in between. Of course, you can cut a paper only in between, right? You cannot cut paper on the edges. How can you cut a edge of a paper, right? You cannot cut a edge of a paper. That is why it has been named endonuclease. The one which cut the DNA is called the endonuclease. The first restriction endonuclease was HIND2. Okay, it was HIND2 whose functioning depended on a specific DNA nucleotide sequence was isolated and characterized by 5 years later. So this was isolated and characterized only 5 years later. And it was found that HIND2 always cut DNA at particular points by recognizing a specific sequence of 6 base pairs. So number 6 is important over here. Thing is that this HIND2, when observed carefully, when studied carefully, it was revealed that this cuts at specific points at specific sequences of 6 base pair. So let us consider there is a 6 base pairs, 1 2 3 4 5 6 and 1 2 3 4 5 6. This HIND2 cut only at specific regions, maybe over here, and yes, this is of course, if this is 5 dash to 3 dash, then this will be also 5 dash to 3 dash. So of course it will cut over here. So HIND2 was observed that it only cut at specific sequence of 6 base pairs. Now this specific base sequence is known as the recognition sequence for HIND2. So this entire sequence is called the recognition sequence. So besides HIND2, we know more than 900 restriction enzymes that has been isolated from 230 strains of bacteria, each of which recognize different recognition sequence. So each of them have different recognition sequence, and totally we have 900 restriction enzymes up to date. Though restriction enzymes are molecular scissors, they are named so because they restricted the growth of bacteriophage. The convention for these enzymes is the first letter of the name comes from the genus and the second two letters come from the species of the prokaryotic cell. So now let us enlarge this name EcoR1. EcoR1. In this, right, the first letter, that is E, comes from Escherichia. It comes from Escherichia. Then CO comes from coli, and then R over here, R comes from the RY13, and number 1 tells the order. For example, from Escherichia coli RY13, if you have isolated maybe 10 enzymes, 10 restriction enzymes, then this is the first enzyme of the 10. So E from Escherichia, CO from coli, R from RY13, and 1 is the order. Okay. So the Roman numbers following the names indicate the order in which the enzymes were isolated from that strain of bacteria. Restriction enzymes belong to a larger class of enzymes called nuclease. Nuclease is a type of enzyme which alters the genetic chemistry of the nucleotide sequence. So those enzymes are called nucleases. And of course there are two types, that is exonuclease and endonuclease. As I said, endonuclease cuts between the paper, right?
It can't cut on the ends. So exonuclease is the other type. So exonuclease, what exactly does? It removes the nucleotides from the ends of the DNA. Unlike endonuclease, which acts in the middle, exonuclease acts on the ends of the DNA, whereas the endonuclease makes cuts at positions within the DNA. So I hope you understood what is the difference between an exonuclease and endonuclease. Exonuclease removes the nucleotide from the ends of the DNA, while this one acts within the DNA. Each restriction endonuclease functions by inspecting the length of a DNA sequence. As I had shown you in the previous slide itself, it acts at specific recognition sequence, right? So in order for that, it has to first inspect, right? If you want to find the culprit, then you have to inspect the big line of culprits, right? So like that, this restriction endonuclease will be inspecting the entire length, and once it finds this recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix, right? Double helix, no, DNA is double helix, so it will correspondingly cut at the specific sequence between two nucleotides at the specific sequence in their sugar phosphate backbones, and each restriction endonuclease recognizes a specific palindromic nucleotide sequence. Now, what is palindrome? Palindrome, I'll explain it in the next slide. Palindromic nucleotide sequence we will see now. So basically the endonuclease will be acting at this palindromic nucleotide sequence on both the sides of the double helical DNA. So guys, now I'm going to explain what exactly is a palindrome. Okay, so most of you would be knowing what exactly is a palindrome, but let me explain it for you with an example. Now let's take an example like race car. Now this I have written from left to right, right? Now if I want to write this again from right to left, let's write again. So from here I'm starting R, then A, then C, then E, then C again, then A and R. So basically, if you read either in the direction of left to right or in the direction of right to left, it will remain the same, right? So similarly, you have another word called Malayalam. Just try reading in the opposite direction, and yes, it will read the same in the both directions. Now the palindrome in a DNA is a sequence of base pair that will read same on the two strands when orientation of reading is kept the same. Now see, now I am reading from 5' to 3' GAATTC, and similarly if I read from 5' to 3', then it will be GAATTC. Now this is the palindromic sequence. Now if you read from 3' to 5' direction also, CTTAAG, and even over here CTTAAG. So this is called the palindromic sequence. So restriction enzymes cut the strand of DNA a little away from the center, meaning over here if it wants to cut, it will be cutting somewhere over here between the G and A. It will be cutting at G, between G and A, and not over here, not between GAA and TTC. It will be cutting between G and AATTC. Okay, so it is little away from the center of the palindromic sequence, but yes, between the same two bases on the opposite strand. Regardless how big the DNA be, how complex the process be, you can rely on your human physiology that it will be cutting between G and A only, and not even little away from the two bases. This leaves singly stranded portions at the ends, and these are nothing but overhanging stretches called sticky ends on each strand. Now if this is left over, so basically you will have GCTTAA as 1, and another sequence will be AATTC and G, right? So you will basically have these regions, and these are nothing but sticky ends. This region is called the sticky ends, and these are overhanging stretches. These are named so because they form hydrogen bonds with their complementary cut counterparts, and this stickiness facilitates the action of the enzyme DNA ligase. So this stickiness is very much comfortable for our enzyme DNA ligase. Their tendency to form hydrogen bonds between the sticky ends is very much favorable for the action of our enzyme DNA ligase. So this is the diagram depicting the action of the restriction enzyme, and if you note over here, the restriction enzyme cuts between GA and GA on both sides, and this is the restriction enzyme, that is eco R1. Once this cuts the enzyme, right, you get this sticky end over here and this sticky end over here, and these two tend to form hydrogen bonds, right? These two tends to form the hydrogen bonds, and this tendency is the one factor which is favorable for the DNA ligase, and these fragments join at the sticky ends to form the recombinant DNA. So you can clearly see in this diagram that the enzyme cuts both the DNA strands at the same site, and over here it is important to note that it will always cut between G and A. Okay. So restriction endonuclease are used in genetic engineering to form recombinant DNA molecules which are composed of DNA from different sources or genome. When they are cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and yes, these can be thus joined end to end using DNA ligase. So it is important that both the plasmid and your piece of DNA is cut with the same restriction enzymes. It is important that you use the same restriction enzymes. So unless one cuts the vector and the source DNA with the same restriction enzyme, then you cannot create the recombinant vector molecule. Okay. So this is another diagram which shows that you are basically cutting with the same restriction enzyme. That is, this is your foreign DNA and this is your vector DNA, that is the plasmid. And when you cut, you get the cut ends over here. And when you use ligase, this piece of DNA is incorporated over here. And then there is a process called transformation wherein this plasmid is introduced into this E. coli cell. Okay, transformation is a process wherein the vector, the Rdna, is introduced into the cell of the E. coli, and when that divides, of course, you can see this happily over here as well. Okay, this is something which we have done in the third slide, I guess. So since you have incorporated it into the specific genome which has the ORI as well, you can expect the foreign DNA, that is this blue color foreign DNA, in these progeny cells as well. So alright guys, that's the end of the video. If you do like this lecture, please subscribe to my channel, please press the like button if you like the video, and please share this video to all those if you find this video as useful, and you can leave all your comments and suggestions in the comment section below. So alright guys, thank you so much for your support.